registered students, how many of you had access to the query system now? Show me your hands. Okay. How many of you were not able to get in? What will happen for the people who didn't show hands in either time? You haven't tried it yet? All right. So it seems that the query system is working, not on the Windows system, and for some reason I have no idea. So if you can, if you're using Windows and trying to get access, and probably want to contact UITS for the help. All right, we just tested here, and it works on the Mac system, and it also works on the Linux system. All right. Okay. Another thing I want to mention today is for next lecture. Next week I will be out of town, so for the next lecture, and it will be a guest lecture from the Center for Medical Genomics, and you will need to go to BRTC for that lecture. So that will be about half of the lecture will be talking about the sample preparation, for the library preparation part, biochemistry part of it. And the second part will be the tour to the sequencing facility. So you will see how fancy the machine is, what it looks like, okay? Why doesn't work, or things like that. So, and that will be exciting. But I apologize for people who didn't register class, and the classroom over there are extremely small, and they can barely host the registered students. So next, it's a break for you guys. So next week, all right, for the students who didn't register. So everybody know where the BRTC is, right? So there's no problem to get there. All right, ask your neighbors if you don't know where that is. And another thing is I just gave another talk this morning. So if I start to lose my voice, but towards the end of today's lecture, just bear with me. All right, so we had 2 lectures in the previous 2 weeks. And the first one was a brief overview of everything, right? Next generation sequencing, how great the technology is, what the different platforms are. And the second one is really going to the data processing part, including the alignment and the data format and those kind of things. So from today, of course, I know I have a little bit left over last time. We'll still finish it. And from today and the next 3 lectures, not counting the next one, we'll be talking about DNA sequencing. So today, I'm only going to talk about so basic concept of SNPs and those kind of things, and what type of genetic variants are available. And I'm going to introduce several published studies that are very successful in terms of DNA sequencing field. For the next lecture, 2 weeks out from today, and I will be talking about the bioinformatics issues related to the DNA sequencing project, including the mapability and the structure variation and how to identify genetic variants and all those things. And then the third lecture of DNA sequencing, the beginning part will be Dr. Tatiana Freud will give you one example of how to use this technology to study specific disease. And then the second half of that lecture will be some leftover from the bioinformatics part, including how to prioritize the variants and those kind of issues. Okay? So just a brief overview of last time. So the data analysis for the next generation sequencing, including 3 components: the primary analysis, secondary, and tertiary analysis. The primary analysis is basically for the data acquisition and the change to the base call. Usually, we don't have to worry about this part because the sequencer itself, the machine manufacturers, hopefully they will do a good job to take care of this part. And the secondary analysis are the bioinformatics part that are not really specific to certain biological question. So it's universal thing, including sequencing alignment, statistics, consensus calling, and create QC files and tag. And so basically a sequence assembly. So those are considered as the secondary analysis. For the tertiary analysis, And this really need analysis team to work on that, because you need to know not only the bioinformatics part, how to do the programming, but also you need to know the biological question that being answered. And it's very specific to different applications, even for, let's say, ChIP-seq. And there's depends on what kind of problem you are facing on. If you study transcription factor or you're studying DNA methylation, it will be different analysis pipelines. So those are very specific to individual project. And this part will be the analysis team that is should be working on that. And still, a review from last time. So we mentioned, we keep talking about the quality of the data. And there's a couple of different qualities that are mentioned. The first one is the base quality. So basically, is for each individual variant sequence, and you want to say this is AATT, and how confident you are that that particular base, the sequencer did a good job. So that is the base quality. And the second level quality is the so-called mapping quality. So what happens is for each individual sequence reads here, and you want to map it back to the reference genome, okay? Human genome, for example. And what is the probability that your alignment is correct? And that is referring to the mapping quality. The third level is called the consensus quality. And this part is specific for each individual variant being identified. So if there is, you say this is a homozygous alternative or heterozygous, what's your probability that is right? So this is 3 levels of qualities. If people refer to give you some data and show this is the quality of it's 30 or 40 or whatever the number is, you need to know what quality are they talking about. It's a very important concept. And we also very painfully reviewed alignment algorithms. So there's always, if you talk about anything related to alignment, and probably Smith-Wortman is something that you have to go through, although we know that nobody is really using it, all right, because it's too slow. And for the fast alignment algorithm, so we mentioned there's several different categories. So most aligners either use hash table based. So basically, you build a dictionary of the sequence rates, and then you go back to the dictionary to find out where that is in the genome. Or the suffix tree or prefix tree based, and we know there's something called the Burrows-Wheeler transformation, and which is very, very smart. Okay, and this figure shows that the resource that being used, if you use the BWT, and actually the size of your index is only 1.65 times of the genome size that you are working on. Where for the dictionary hash table based, it's 15 times, and for suffix tree based, it's 45 times. So those are so in terms of this, in the exam, I'm not going to ask you to tell me how to do Smith-Wortman or how to go through the Burrows-Wheeler transformation. Rather, the reason I talk about this is I want you to be able to know that what type of strategies people using, because there's 24 of aligners already there. So you want to know, as you pick out an aligner, or tomorrow maybe another aligner published, and you want to know that which category it fits into, whether you like it or not. You can have your more intelligent judgment on that. So there's no reason to be panicked if you cannot go through this, how to do this kind of calculations. Rather, you want to know the basic concept of it. Okay? And the last part we talked about that was the data file format. And we know that this is a very painful, very dirty work. You have to really go back to the lecture notes and to see that what the data look like. But basically, we talk about 3 levels of data. The first one is the so-called FASTQ data. So the thing in the square, they are standard format. Okay, the FASTQ format is just off the sequencer, and it gives you the ACGT and all those sequences, and together with the qualities. And those raw sequences are FASTQ file. And after you're doing alignment, so those the sequence rates, you need to align it back to the reference genome. After you do the alignment, the data format is called SAM file. Okay, and these are the things that you really need to, I'm referring to the student who register, really need to go back to know that what formats, what information is stored there and how you want to process it. And so we stopped last time here. And now I'm going to go through a little bit about the VCF file, which is the variance information here. Okay? And this VCF file is also first developed in the Southern Genome Project. So you see, the Southern Genome Project is a really big deal. It's not only the scientific question help you to identify the human variants, it really generates a lot of standard for the data processing. And the standard format storing most prevalent type of sequence variations, including SNPs, insertion deletions, and large structure variations, together with very rich annotation. It has these two components. One is the so-called Vcf headers, and those are really some of the information that is documented in this file. And from here, these are individual variants, and we will go through that a little bit in more detail. Okay, so the first one is so these two. For the type of variation that VCF file will document include the following, including SNPs or deletions, and large structure variations, insertions, and other more complex events. So you can see this has the field to give you what the reference nucleotide is and what is the alternative nucleotide is. The alternative are the variants that are already being identified in that particular location. And also there's several columns regarding the quality. So you can see this quality is really the variant quality. So how strongly you feel this, how accurate you think there's a variant documented in that particular location. And again, this is somehow it's a Fred scale. It's a minus 10 times log 10. And Fred scale is very popular in the sequencing data analysis. So there's a small concept that you really want to get familiar with is if there is a variant there, okay, if there's a variant there, the quality really refers to the p-value of there's no variant, okay? So if there's no variant, and if there's no variant, what's the probability that you detect something? So what's the probability you made a mistake, so basically. And here is if there's not a variant detected, and this is really the probability that there's a variant there. So always in this p-value are something that what's the probability you made a mistake, simply put in that way. What's the probability you made a mistake? Let's say if the probability you made a mistake is 0.001, that is pretty good, right? So, and you made a mistake, it's only 0.1%, and this quality value after you do the calculation will be 30. So in other words, the higher the quality score is, the less mistake, and simple as that. And also you will see there's a filter column. So the filter column gives you the information whether it passed, so if all the position pass all the filters in the header files, or which specific filter has not been passed, has been failed. So the reason is that you will see that in the header files of this file, there are several filters that will be defined. For example, if I said ID is Q10, that means the quality below 10. I want the quality is better. So if it's quality below 10, it will put a Q10 here. That tells you it failed the passing the filter and what the reason is. So all this information will be in the header files. And the next group of information are additional information for this particular variant you identified. And it's optional. There's 18 predefined options, but very important ones include the database, which means the DB-SNP membership. So remember, if you identify a variant, it may be a new variant, or maybe a variant has already been documented. If it's a variant already known, it's very likely it's already documented in DB-SNP, and this is a good place to document those information, okay? And the DP is combine the depths across all the samples. So what that means is if you see there's a variant in one genomic location, it's not going to be supported by only one sequence read. It usually is multiple read and stacking on this area so that you know there's a variant there. And here, the DP simply tells you what's the sequence depth that is located in there, and to support this variant. NS, numbers of samples with data. Okay, so because there's multiple samples in the later columns, and how many of samples that has this variant has been identified. AF, estimate allele frequency, so it's very straightforward. And strand bias, whether there's a strand bias for this position. And I'm going to go into a little bit more about this in the next lecture. And AA is ancestor alleles, whether it's in chimpanzee or things like that. And so those are the information that is more specific to this particular variant being identified. So when you go back to review your lectures, so you will know that all this information are documented in there. And you may think, oh, I don't really need that information. I only need there's a variant. Okay, it's never that simple. So when you really get dirty into processing the data, and those are the information you need to pay attention to. And those are in the VCF files. And the next couple of columns are really for each individual samples. So this part I was just talking about is for this particular location. It's a summary information for all the samples. Now, you can get into individual samples a little bit more. And so basically, the data fields have some type of data should be presented for all the samples. So the format defined in this part, okay, it tells you the genotype. And if the genotype is 0, if it's referenced, that will make a 0 there. And if it's first alternative or second alternative, because some variants may have one alternative allele, others may have multiple alternative allele, so the options are there. So this, if there's this type or this type, that are the genotype unphased or genotype phased, okay? And GQ is a conditional genotype quality. Again, this is another level of quality. So it's a conditional quality given if the variant does exist, what's the probability that the genotype you called is wrong? And again, the higher the quality is, the more accurate your data will represent. So those are the information are all documented in these fields. DP, read depth at position in this particular sample, not overall. And haplotype qualities. And all those informations are in this in these fields. So actually, this is much more complicated than this. And if you go to the OnCourse system and you see we do have a PDF file that has all the VCF file defined. I think it's close to 100 pages. It's a pretty massive documentation. So if you really, this is just to give you a crack of it. So if you want to go to understand what the variance information look like, you need to go to that website and to take a look at it. Okay? And the VCF files can be complicated. So I mentioned the SNPs, insertion, deletions. Those are simple stuff. Okay? What if the DNA is really sick, like in some cancer type of genome, and they become recombination, things like that? And the VCF files can really take care of this by looking into all this. I'm not going to go through that because that will make me dazzle. If you think this is not complicated enough, and they even can, this VCF file can even describe this type of recombination and the structural variations. Okay, and I don't want to get into it. Okay, so we talk about the standard data formats. The QSeq file, which is the original raw sequence, SAM or BAM file, which is the alignment information, and the VCF file, which is the variant information. Okay, so you can go back to look at that. So still, there's some information regarding data visualization. So we know that the data is massive, right? Several terabytes of data, and after alignment, there's half a terabyte of data. How do we really see where the reads are, okay? And the first two that I want to introduce are genome browsers. How many of you have used the Ucsc Genome Browser? Okay. How many of you have not used the Ucsc Genome Browser? All right, there's a few. So we'll get into this a little bit. And so there's many open resources and public resources to facilitate exploration and biological interpretation of genomic data. And Genome Browser can bring together genomic data and additional annotation data together. Okay? And Genome Browser will provide the context for the chromosome locations and search or navigate genetic regions, especially when you have your own data. Okay, there's a multiple type of genome browsers, actually. And when we talk about genome browser, we always refer to Ucsc genome browser, actually. But there are several types. And specific, there's a linear genome browser or circular genome browser. And this circular genome browser can be very interesting, sometimes useful as well. It's just for describing some other type of data. For the linear browser, and there's a EBI, Ensemble Genome Browser. You can go into look at that. It's very useful. And NCBI has a map viewer, which is also part of the genome browser. But the most important one is the Ucsc Genome Browser, which we will talk about today. Here's the Ucsc Genome Browser. I didn't make these slides, and these slides were in the Ucsc website. And if you want to download, you can go there as well. Okay. So this is when you go to this website, and this is what shows up. So there's many different tools, actually, but the only information, there's a general information here, specific information, and the navigation part. But the only information we need is you click here, right? We go to the Ucsc Genome Browser. And the Genome Browser, this is the gateway start page. Okay, you can see that you can really select the species that you want to study. It's a mammal or human. You can go through many different species, okay? And use this gateway, you can search gene name or gene ID or genomic location or types. It's very, very powerful. And these are, they do have something to tell you what type of query that you can put into here. And the first thing you can look at is you need to select the clade, whether it's mammals or insects or what different clade it is. And once it's selected, and you want to make sure that you are in the same species, right? If you study human or mouse, and things can be different. And even for the same, this is important, even for the same species, there are usually multiple assemblies there. And we have seen very common mistake that people say, I know this genomic location has some information, but I go to Genome Browser, it's not there, okay? Because you're using the wrong genome build, okay? This is very important. It's a trivial issue, but that's the common mistake people make. And there's the latest one is HG19, which is published in 2009. And in this part, you can really put in the information you want to type in, either the chromosome locations or either gene name or things like that. And you can even specify how wide your viewer you want to look at. Okay? And by this, you can select the tracks, or you can configure multiple viewing information. And this is one of example here. So you can put the P53, just type the gene name in and click submit. And this will be all the genes related to P53 will be show up here. And you can select the results you want. Usually what I do is I'll just randomly pick one of it. And this will be the entire overview of the whole genome browser. Of course, this is not going to be the view you are looking at. But the point is, this top part is a genome viewer session, and the later part are just a different type of annotation tracks. And it's very rich information, and including the genetic variation, the annotation of the gene. So you can really go there to take a look at. All right. And for different species, you always see the same pictures, but actually they are different. They have different tracks and using the same software. For different species, some species, for example, human, you have DB SNP information, gives you the variants, where documented variants are. For the mouse, you probably don't have it. So it's very common that you will see different tracks in different species. Okay? And Again, this is just one of the example, T53, and type in the genomic location, and then you can see that all these different annotations are there, and this is the exons or introns, and all those information are listed there. And there are some, the 44 species compared, the conservation information, a single species compared, or SNP information. So all different type of annotations are listed there. If you're interested, just go to try it. Okay, now, most of you already know Genome Browser. So what I just talked about in the past 5 minutes, it's straightforward. Now, there's something that I want you to know, okay? So in the Genome Browser, if Genome Browser only designed to see the annotation of the existing biological information, it's not very useful. The powerful thing of Genome Browser, it will allow you to upload your own data into the Genome Browser and overlay it with the existing annotations. So if you study ChIP-seq, you identify a peak in a certain genomic location, you want to know, is this peak in the promoter region of a gene? And what is that? So you can load your data into it and then overlay that with existing annotations. That's very, very important. There's two types of files. So there's actually a button, just add custom tracks there, and you will see a picture like this. That is straightforward. Just go to take a look at. But there are something that even people use this tool a lot didn't realize. There's two types of files you can really upload. Okay, one is the text files, and if it's a text file, including a BAT file, GFF, and Wiggle file, and all those information. And you can just put your file name in here. You can really upload it, okay? Or if your file is a binary file, this binary file usually are large files, including the BAM file, for example, which remember is the alignment after the binary format of SAM. And those are really, really very large files, several terabytes. You do not do that. You do not really load the data into the Genome Browser. Rather, you only provide a link in here, and then you still put your data somewhere on your server so that when you do the query, and what happens is you say, I want to look at this genomic location. The Genome Browser will go to your server to search the data and send it back to Genome Browser, and then you can see it from the web browser. So that is how it works. And just to keep in mind, if the file is a text file, direct upload. If it's a binary file, and provide the link, okay, Genome Browser will know it. Okay? This sounds straightforward, but I see people in my lab are making mistakes. That's why I talk about this in greater detail. So these binary files include the BAM files, big BAT, and the big Wiggle files. So if you want to know the definition of those files, and go to the Genome Browser website, they do have it. Okay, and another thing I want to talk about is these different viewing options. So you can. There's really several, so for each track, there's several possible viewing options. If I just put it there, and hide, that means there's no data, of course. Dense, so you will see the data that look like this. All these things are the exactly same data, okay? And squish are like this. So this is the one that I prefer the most. So if I want to put some kind of snapshot in one of the publications, squish will be the one that I use. And there's a pack, or full. Full is really, you will see the data going very, very long. So you can see that this is probably one of the sequencing information, the start and the location, and things like that. Okay? And so much about Genome Browser. So Genome Browser is useful, but it can upload your own files in there. There's a difference between text and the binary files, and there's a different viewing options. That's the information I want to deliver. And there's another Genome Browser which is very useful. It's called Integrative Genomic Viewer. And so especially for the students who register this class, and that will be in the next homework. Okay. So the first homework, remember, we are talking about setting up query accounts. Okay. Once you get all this passed over, and then I will give you some real data, and then you try some existing alignment algorithm. So we will generate a BAM file, and then next stage, I'll ask you to really view these BAM files, and using Genome Browser and the integrated genomic viewer, both these two softwares. So probably it's a good time you install the software if you want to bother your administrative people once, anyways, right? And this is a Java-based program, and it's not web-based, it's Java-based. So you have to do some installation in your own computer. Okay? So Genome Browser and IGV are the common tools that in my lab we're using to view this data. All right. For this lecture, those are the leftover from the previous ones, the data formats and the data visualization. For this lecture, I'm going to go into a little bit more basic about genetics. And actually, I wasn't trained as a geneticist, so I will just present whatever my understanding is. And Ken, you can correct me if I'm wrong. Oh, you are not? Okay. That's very good. That's very good. I like it. Okay. And after that, I'm going to present a little bit on the states of genetics of complex disease before we have next generation sequencing technology. Where were we before this technology? And then I want to introduce some kind of real impact this NGS technology enabled. And the first one is the NGS and the genetics of complex disease, and trying to understand the cause of the disease and how we use this technology. And the second most important part, I view this as the most important part, is the implication on the personal genome sequencing. And we know that for the next 10 years, it's probably very important for personalized medicine and personal genome information. And this, without this information, everything is just not there. And in the end, I'm going to present something regarding the experimental considerations on the DNA sequencing experiment. So today is all about biology. There's no equations, I promise that. Okay, genetic variations. So there's a different type of genetic variation. The first one is a SNP, a single nucleotide polymorphism. It's 1 in every few hundred base pair, and the Southern Genome Project actually concluded there's about 1 in every 300 base pair of SNP. And we will have more definition on the SNP in the later slides. So the concept is quite simple. It's just a 1 nucleotide mismatch, right? And the mutation rate is 10 minus power minus 9. So what this really means is for every generation, for every base, what's the probability it will change to something else? Okay? So if you think of in human genome, if we have 3 billion letters, so there's about 3 mutations for every generation that is generated. However, if we're talking about the developmental stage, there's multiple things. I mean, cells, we need to do the duplication and those things. So each part that then this race also applies. So it's not really for 1 generation because we are a multicellular species. Okay? The second one is short insertion deletions, and these are considered a simple type of genetic variation. And it's also in 1 in every few kilobase, and the mutation rate really varies. And this is one example. If this is the reference genome, this is the sample you are sequencing, and you got 3 more letters comparing to the reference genome. This is called a small insertion, right? And another type of genetic variation is the microcytolytes and the repeat numbers. And this happens in 1 in every few kilobase. And actually, those repeat sequences are usually 2 to 6 base pair repeat units. It happened many times, okay? And in the earlier genetic area, people use this as a microcytolytes as a genetic marker to study which part of the chromosome are associated to a certain disease. And a bigger scale, there's something called the mini-cytolytes, not microcytolytes. And the difference between these two, the mutation rates are different, but the major difference is that their lengths are different. So if you see the repetitive elements for the microcytolytes are 2 to 6 base pair, and here is about 10 to 100 base pair. Okay? And there's many other types, and including the repeated genes, the ribosome RNA, histones, and so there's an even larger scale, and also the larger structure variations, including the insertion, deletion, duplication, inversion, copy number variation. We kind of see this a little bit in the first lectures. Okay, come back to the SNP. I want to give a more introduction on the SNP because this is the most common type of variation that we will observe. So a SNP is defined as a single base change in DNA sequence. Okay, that's simple, we know that. And that occurs in a significant proportion, more than 1% of a large population. Remember I mentioned the Human Genome Project tells us 1 in every 300 base pair there is a SNP. But if you talk about other type of single nucleotide variation, that occurred much more often. Okay, when we talk about SNP, it has to occur in more than 1% of the population. And okay, 1 in every 300 base pair in human genome and 10 million SNPs across the genome. Okay, that doesn't mean that one individual has 10 million SNPs. Okay, that's a human as a whole that we have about 10 million SNPs. That's the current estimation at least. And SNPs are conserved across the genome, often in patterns called haplotype blocks. So the reason I want to talk about this is I want to give you the introduction of what is haplotype in the later slides. And SNPs are the most simple form and the most common source of genetic polymorphism in human genome. 90% of all human DNA polymorphism are associated with SNPs. Important. All right. There's two types of SNPs, and this is kind of important. And there are two types of nucleotide-based substitution resulting in SNPs. One is called translation. The other one is called transversion. Okay? For the translation, that means substitution between purines or between permitins. So if the reference genome is A and they become a G, that is a translation. If it's C become a T, it's a translation. Well, transversion is a substitution between a purine and a permitin. If it's A become a C or become a T, that's a transversion. Does that make sense? Okay? All right. And the reason I think this is a big deal because I didn't know this not a long time ago. So I just want to make sure that everybody is on the same page because I wasn't, okay, a couple weeks ago. So the ratio of the number of translations to the number of transversions for pair of sequence becomes 0.5 when there is no bias towards either translation or transversion substitution. Okay? So the reason for that is if you got an A, if it would reference is the A, you become a translation, you can only become to a G, that's a translation, right? If it's a transversion, you can become either C or T. So you can double the chance to get a transversion if there's no bias at all, okay, between translation and transversion. However, recent human studies, particularly from Southern Genome Project, have shown that for the whole genome, a translation-transversion ratio is 2 to 2.1 is generally correct. In exons, this number goes to even higher, goes to 3. Okay, and you may think this, how do we use this type of information? And actually, this is a kind of evaluation for the quality of variation you identified as a whole. Okay, we'll talk about that next time regarding how to use this translation transversion. But the concept here is simple but important. Okay, SNPs are not independent events. So if we talk about two SNPs, okay, they are actually linked together. Okay, now brings in one concept called allele. Okay, allele is one of two or more forms of the gene. So there's a, okay, I'll use this one, it's closer to me. A, and B, and each one is an allele. Okay, And the recombination leads to the generation of new combination of allele. So you can see that in the original case, and this is the ABC all small and the big ABC all small, right? And then it duplicates, it becomes the same. But if this part recombination or crossover happened here, and then you're generating a new combination of alleles, so it becomes small AB and large C. Okay, here is a big, large AB and a small c. So there's a new combination will be generated. So this is the result of recombination. And the groups of alleles located close to each other on chromosome are rarely separated by the recombination. So what happens is if this small a, b are pretty far away from this c, okay, and when the recombination happens, it's easier to generate this type of pattern. But if it's very rare that you will see that A, B are in different recombinations. So that only happens when the recombination happens in between of this, and that is kind of a smaller probability comparing to happens somewhere here, right? Because it's a larger area. So what happens is neighboring SNPs are frequently inherited together. They are never, I shouldn't say never, they are rarely separated. Okay? So the point here is SNPs, they are not independent event, rather they kind of inherited together, especially for the neighboring SNPs. And this will come to the concept of so-called haplotype. So you can see the haplotype are actually the blocks of associated SNPs. Okay? If we know that there are some SNPs, they are pretty close to each other, and the combination of these individual SNPs will eventually form a haplotype. Okay? Blocks of associated SNPs together. Okay? Is that something understandable? All right, and this is haplotypes sometimes refers to different phenotypes. So you can see one example here is the eye color for that particular gene and different colors. And the black eye and this 3 SNPs is A, G, or deletion here. And that will become a black eye, and the brown eye is GTA, and the GTA. So those different haplotypes can refer to generate different type of phenotypes. Okay? So we're talking about alleles, which is the individual variants. Okay? We're talking about the alleles, their recombination can cause the neighboring ones to always inherited together, and that they form as haplotypes. All right, structure variation. Next type of genetic variation. So traditionally defined as deletions, insertions, or inversions larger than 1 kilobase. And they are often involved in repetitive regions of the genome and complex rearrangement. So you can see here, this part, there's ABC 3 chunks, and deletion here is the middle part, the B is missing. So we kind of see this quite often in the previous slides, but there is a general separation between the small insertion deletions and the large structure type of insertion deletions. The reason is for the smaller ones, if the insertion deletion only happens a few base pair, and the end result of that is you're losing one binding site of transcription factor, or you are increasing one amino acid into the protein. So there's an important and sometimes serious changes, but you still result into the same type of proteins. But for the large insertion deletions, and you can see there's one big chunk of genomic region are missing. So you don't get that protein at all sometimes. And also the insertions. So in the original genome, we got A, B, C. Now there's a big chunk D is inserted here. Okay. Inversions. So you can see the order of this is A, B, C, and the inversion of this will become C, B, A. Okay. Somehow it kind of shifted the directions. And copy number variations, and this A, you got multiple copies. We know it's very important, and sometimes it causes, in cancer, there's a lot of copy number variation has been discovered. And also segmental duplications, A, B, C, another ABC there. So there's all different type of structure variations. And another reason that we separate large insertion deletion and small insertion deletion into the is for the next generation sequencing, we know the read size is not that big. It's a 50 to 100 base pair, right? If there's a 1 or 2 or a couple of nucleotides missing in the middle, and with the adjustment of alignment algorithm, you can still identify those. However, if the insertion deletion happens 1,000 base pairs, and when you do the alignment, it will become really, really, really difficult. Actually, before we have next generation sequencing, there is really no way to identify, no best way to identify this type of structure variations. I shouldn't say no. There's some kind of fish type of assays, you can still do it, but it's very difficult, especially to identify the novel ones. But with the next generation sequencing, especially as the design of a pair ends or mate pairing, and you can make this possible. So if you don't remember that, refer back to the first lecture, and we have a couple of things to talk about how to use pair end or mate pairing to identify the structure variants. And structure variants affect a larger fraction of the genome as compared with SNPs, which is only 1 nucleotide. And the analysis of 1,000 Genome Project identified a total of 1,700 structure variations that will affect encoding sequences. Okay? Keep in mind, in 1,000 Genome Projects, those are healthy individuals. Especially, I mean, at least they are healthy now, right? And but there are so many structure variations affect their coding sequence. It's kind of shocking results. Okay, that also tells us how important these type of things are. And once a bigger chunk of fraction of genome are changed, it can have more dramatic effect on phenotypes. Okay. But why we study genetic variations? Of course, we have to keep the job, but there's some real meaning for that. So SNPs can serve as a genetic marker to identify genomic regions associated with disease, okay? Which is nice. So that means that it's in contrast with some SNPs will cause the disease, okay? And these are, at least they can serve as the genetic markers to identify genes that are associated to disease. And disease-associated SNPs, regardless of their functions, have potential for clinical applications, and including predictions of disease risk, treatment response, and prognosis. Okay? That doesn't mean those SNPs are causing this difference of phenotypes. At least they can serve as a marker for those. Some other SNPs may be responsible for abnormal gene expression and protein function that drive disease process, and these are generally called causal SNPs. Okay? So there's even for the non-causal SNPs, they are pretty important as well. And most genetic variants have no effects. Okay, we talk about in human, we have about 10 million variants. So for one individual, you probably have 1 million SNPs in your genome. Okay, most of them, genetic variants in the human genome, are silent variations. So they are not important. They are not having phenotypic effect at all. And they do not occur in coding or regulatory region of the gene, or are within these regions but no effects. So again, not important. But they are still very useful. And in some, they have some specific applications like forensics, okay, and paternal test, and archaeology. Okay, there was a paper published last year. I don't know whether you see that Nature paper. They have this, they made this cover face of the Nature, and it was a guy, ancient people. So it says ancient human genome. And so the story was there's one guy had in Greenland, I think, got a haircut about 4,000 years ago. And they identified those hairs and they sequenced it. And so because of that haircut, we know him much more than he knows himself. Okay? And he's predisposed to some kind of cardiovascular disease and all those things. I don't know whether that happened or not, but that's one of the applications that people can do, right? There's another, just saw the news yesterday, and there's one other Chinese story, is you know that, especially for the non-Chinese students there, those kind of 2,000 years ago, the first king of China has big tombs, and a lot of people from all different kind of area size of China and come to build those tombs. And they were trying to see who are those people, okay? And then they identified some samples, and they did the sequencing, and they concluded it's more workers are from slaves from one part of China versus the other, and how those migration happens. This kind of interesting stuff, right? And but anyways, even for those genetic variations that there's no phenotypic effect, it can be useful. However, in biology field, we always want to go to the functional SNPs, right? These functional SNPs, including the nonsense SNPs. So I will try to go through all these things, right? Including the nonsense SNP. So the so-called nonsense is within an exon and translated amino acid change to a stop codon. All right, that is a big problem. So you think of there's a gene and the RNA, whatever, and transcribed, and then you do the translation. All right, and from beginning to the end, but not to the end. There's somewhere in the middle. There's one mutation happened, and it becomes stop codon. So translation doesn't further go along, and that is a much shorter gene, right? And the protein, and it will eventually trigger some other effect like nonsense-mediated decay, and it will eventually chew up that protein. And this is really, really big deal. Nonsense mutations are really problematic. And there's some other mutations, variants cause frame shift. And of course, these are because of insertion deletions, right? So think about that. Every 3 nucleotide will become an amino acid, okay? If there's one nucleotide inserted into somewhere, happened there, and it's not only affecting the local area, so all the other frame shift will be messed up. So that gene is no longer functional as well, right? So if you see some insertion deletions happens at 3, 6, and a time of 3, and that would be more mild. It's just 1 or 2 amino acids were inserted into that. It's not big deal, but for the frame shift, that's really problematic. Coding non-synonymous variations. So what that means is you got one nucleotide shifted, change it, and it will change to another amino acid, okay? And sometimes it can be important, others it's okay. So those are called non-synonymous variants. Okay, splicing 5 or splicing 3, this is in the first 2 base or last 2 base of the intronic region. So those are messing up with the splicing mechanism, and then therefore there may be 1 exon are missing because of it, or just 1 exon were included because of it. So those can have very dramatic phenotypic effect. And for the coding region, synonymous variation, and that can be important sometimes. So you can see within an exon I translated, there's no protein amino acid changes. I know that in most genetic analysis, those synonymous variations will be ruled out. We think of this not important and things like that. But it can be important because the RNA binding protein can bind on that, so that 1 nucleotide change may change the binding affinity of RNA binding protein, therefore affecting the further processing of the gene. So that can be important. And mutation can happen in the 3' UTR or 5' UTR. UTR refers to the untranslated region. So some people think it's not important. But now we know that in the 5' UTR, there's a lot of things that control the translational process. Okay, there's a protein will bind on in the 5' UTR and control the translation. If that mess up, and there will be problem. And 3' UTR are also a place that a lot of RNA binding protein, especially microRNA, binds there. So if you have a variation in the 3' UTR, that can have a big problem as well. And near the gene 5 or near gene 3, that refers to the upstream of a gene or downstream of a gene. And those are generally, especially upstream, is the promoter region. So those transcription factors will bind there. If there's a variation there, maybe the transcription factor cannot bind there, therefore affect the gene expression. And we will go through all these things in the 3 lectures from now. And the intronic region, and this can be important, sometimes affect the splicing. Okay? All right, let's take a 10 minutes break. Okay? And we'll talk about states of genetics of complex disease before we have high throughput sequencing technology.